All right. Shall I start doing a little housekeeping for everybody? Waving my arms with the white background. My name is Carol Hamilton. I know many of you, but not all. I am the Senior Director of Development here at the ALS Therapy Development Institute, ALS TDI. And I will kind of be your moderator a little bit today, kind of like the boss, right? I'm going to boss the boss around on this call. Thank you very much for joining us. First, I want to also thank our sponsors for the ALS Town Hall series. And this is a series. So after you get off today, you can go back onto the Town Hall page and see some of the other really great and informative and educational and compelling topics that we've discovered, that we've discussed with the help of the community. Very interactive. So those sponsors are Cytokinetics, Biogen, Mitsubishi Tanabe Pharma America, Orphazyme, Apellis, and Alexion. They are helping cover some of the costs so that we can bring all of this to you. Now, we are going to keep you on mute during the presentation. Hopefully everybody knows how to unmute themselves if the time is right to ask a question, if we prompt that. I recommend that you go in speaker view so that you can see Fernando's slides as well as possible. And as you probably heard when you logged on through a giant recording, recorded voice, we are recording this. So if you prefer to remain anonymous, you can turn your camera off, which some of you have. You can change your name on your little rectangle screen. You can be all kinds of creative if you want in the name that you put on there. So however you'd like to protect any privacy you would like to maintain, feel free to go for that now. I want to introduce some of the people on this call that I have the pleasure of working with, some of whom you may already know or will be hearing from soon. Could the development directors wave your arms and say hello? We have Claire Reach and Glynis Murray, who I think I see waving their arms, and I'm not sure if Terry Handler is on camera, but many of you know Terry. And not only will TDI, ALS TDI, be sending you a follow-up survey to get your feedback on today's town hall, but you'll be hearing from the development directors to also get more feedback, to reach out if we didn't have a chance to answer your question, all of those kinds of things. And please do, when you get the survey and when you hear from a development director, we want your honest thoughts. We want to make sure that future town halls reflect things that you are curious about or want to engage over. We don't want to spoon feed you this. We want to reflect the topics that are compelling and useful to everyone in the ALS community. So please be honest with us. And if you think we stunk at something, tell us, because we really only want to get better. Today, I am excited for those of you who do not know Dr. Fernando Vieira to get to know him, as I think you do know. He is now our CEO and Chief Scientific Officer, CSO, at the ALS Therapy Development Institute, and he's going to lead the discussion today around ALS research at ALS TDI and some topics in ALS research in general. I will let him introduce himself in a moment. I just want to let you know that we have a number of questions that have already been submitted. I'm receiving some by text and by private chat, even right now, and I'm sure that will continue. So I think we're going to have a robust conversation. We are going to try to keep to our time schedule. And in order to do that, we are most likely going to save questions till the end. We have a team monitoring the chat so that you can put more in there. For any questions that we do not get to answer in this presentation today, of which there will be quite a few, I expect, Again, those development directors, and included in that lot for these purposes too is Mike Shannon, who is in front of the Golden Gate Bridge taking a swig of something, looked innocent, but a swig of something. You may hear from him as well. So if we do not get to your questions today, please do not fret. We will email you, call you, and apologize for not getting to them, and we will get the answers, and we will continue the conversation that way. So thank you for being here and joining us today. And it is now my pleasure to turn the reins over to Dr. Fernando Vieira.
Thank you, Carol, and thank you everybody for joining today. Taking time out of the middle of a Monday to join us and participate in this town hall. It means a lot to me. I know a lot of you personally, but I don't know all of you, so thank you. I hope to get to know everybody over time here. I am going to give a brief slide presentation today, and then spend most of the time on answering questions that have come in and are coming in. I've learned that, you know, participants, patients, caregivers, people in the ALS community do have the best questions, and those questions do often trigger great ideas for us in our own lab and the way we execute our science. So I really appreciate that. I'm going to tell you, I guess, a little bit about myself. I don't love doing that, but I guess that's part of this gig. I'll give you kind of the resume stuff first. I'll start with that. If you don't know, I'm an MD by training. I studied medicine at Harvard. I have almost 20 years of experience in drug discovery and ALS research in general. And virtually all of that has been at the ALS Therapy Development Institute. A lot of the work that I've been privileged to be a part of has been in literally discovering and inventing new drugs, drugs that are either in clinical development right now or are in the process of moving toward it. And I'm really excited about that. And none of this happens in a vacuum. None of this happens by one person. Every single thing I've ever done has been part of a team. And so anything that I've ever accomplished has been because a lot of people have contributed to it. So I guess that's all of that is the reason why I've been given this opportunity, right, to be the CEO and the CSO at ALS TDI. But a lot of people ask me as well why I would want to be the CEO and CSO at ALS TDI. And I'm going to be honest, I never wanted to be CEO of anything, right? That was not something I aspired to be growing up. Like CSO, yes, all right. That's been something that I would say has been a dream for a really long time. Even back to being a kid, I've wanted to lead science. That's something I could imagine. CEO was a different thing. And it's, I'm in this role because it's our mission, because it's the ALS TDI mission, and it's because it's this organization and this team. And why that ended up being so important to me is a bit of a long story, and it's one I don't often like to tell. And I always wondered why I don't like to tell it. I kind of reflected on this over this past weekend on a hike. And I realized I don't like to tell the story because it's largely not my story. And the person whose story it isn't here to tell it. And something about that doesn't quite feel right. But if he's watching, I hope he'll cut me a pass, and then I'm going to do an okay job at relaying it. So
if he were here, he would not cut you any pass.
I know, he would not. That's the thing, right? That's exactly it. He would be teasing me and ripping into me for how I'm doing this, right? So his name is Sean, and I'm going to get to him in a moment. But when I joined ALS TDF in 2001, it was Therapy Development Foundation at the time. I didn't know much about ALS. I knew about as much as you would learn if you Google it for the first time. And I would bet that everybody who's on this call had a moment where they Googled ALS for the first time. And that's a relatable moment. But for me, it was in the context of looking at a new job in research. And I didn't know much about it. I knew it was devastating, and I knew it was fatal, and I knew I wanted to matter, right? I wanted to do something that would matter. And this place was scrappy. It had a lot of scrappy people who who wanted to aggressively address the needs of people living with ALS. And they wanted to because a lot of them had ALS, or they had somebody living with ALS in their family or in their life. And that was impactful to me, and it drove me into the organization. It made me want to be a part of it. And I quickly got to know a bunch of people, and you know, some of them are still friends I work with today. But one in particular was a gentleman named Sean Scott. And Sean, he was whip smart. And we were kind of drawn to each other. I think we're both super curious. We're both workaholics, I'd say. He had a darker sense of humor than I do, but I enjoyed it very much. like it kind of brought that out of me in a way that was probably healthy. We would just kind of obsess over the ALS problem. And his reason for obsessing over the problem was the fact that his mother had the disease. She had been diagnosed with ALS about a year prior. And his grandmother had also died of the disease, and his whole family had been ravaged by the disease. They were a SOD1 family. He became my close friend, and so his mission became mine, and I desperately wanted to help him help his mom. And so over time, he kind of became like a second older brother for me. He taught me how to play tennis, taught me how to play chess. He kicked my butt at both regularly. We became roommates. We would play video games together. We took vacations together, and ultimately, like, we were family, right? And we were family with a joint mission, with this common bond that felt a bit like a war that we were at together. And there were casualties, and excuse me, he lost his mom. And then it got really weird when his mom's own doctor, right, Dr. Richard Olney, his mom's neurologist, also ended up being diagnosed with sporadic ALS. And so we were all watching as other people that we knew and loved were suffering from ALS. And Sean never wanted to know if he was a carrier of the mutation. But we always were worried that shoe was going to drop. And so we worked, and we had every belief and intention that we would get there in time for Sean. Excuse me. In late 2007, I beat him at a video game, which didn't happen often. And he commented that something wasn't quite right about his hand. And it wasn't long after that, a few months after he was diagnosed with ALS himself, we confirmed in our lab that he carried a mutation, the SOD1 mutation. And it was 9 months after his formal diagnosis that he passed. And so I promised him I would do everything in my power to change this outcome. for people with ALS. And there is literally nothing more that I can do in my power than to be the CEO and CSO at ALS TDI to change this outcome. So that's why I do this job. And that's why I'm committed to it. I apologize. I hate telling this story, and I'm never doing it again for all of you watching. So you've got the recording. So with that, I want to tell you about how we intend to go about solving this problem. With the help of the entire ALS community, there is a process that can be executed that will deliver treatments. And we're executing that process. I want to tell you more about it, and then answer questions about what's going on in the ALS space and more about what we're doing at ALS TDI. Thank you for your patience as I got through that. So I think I just laid it out, the problem, right? We all know the problem. ALS is a fatal disorder, and currently treatments are completely inadequate to the need. And we know that ALS isn't just a single disease, right? ALS is really a collection of many diseases, which manifest similarly with motor neurons in your brain and in your spinal cord degenerating and leading to paralysis. So we know that because it's multiple diseases, it will actually require many different treatment options to address the needs of all patients, right? It's unlikely, if not impossible, that there will be a single magic bullet that addresses the needs of everybody with ALS. So we have a heavy lift in front of us. We have a lot of work to do. And so the solutions in sort of just basic bullet form are, it all rests in research, essentially. And it's research that should lead to discoveries of new therapeutic targets, new interventions, new drugs, and the invention of new solutions. Where it can't be discovered, we may need to invent something to produce targeted treatments that can address the needs of everybody with ALS. And to get there, we need a really engaged patient community that is involved in the research, right? So that we can understand the different strata of ALS and how to get the right treatments to the right people so that everybody has their solutions. And like I said, there's a process to this. And some of you may have seen a version of this slide before. This is the drug development pipeline, or it could be the drug development funnel. There are different ways that people refer to this, but I'm going to walk through each of these steps, and then I'm going to tell you where we, ALS TDI, fit in, and where some other organizations that are doing great work in ALS fit in as well, and how we all can work hand in hand to really address the needs. But I do want to focus in on how essential our work is. So a lot of people describe fundamental research as basic research, right? I like fundamental research because basic sounds elementary, and there's really nothing elementary about it. It's incredibly involved. In fundamental research, you're trying to understand phenomenon in the universe that we don't quite understand yet. It's the type of science where you're digging in and you're discovering. It's the folks who discovered, how DNA is structured, and that DNA leads to RNA, leads to protein, and all of these fundamentally important discoveries that ultimately can lead to treatments. In the context of ALS, fundamental research or basic research often is the discovery of targets, of therapeutic targets. And that leads, once you've discovered targets and validated a target, that leads to drug discovery. In the drug discovery step, you're typically building experimental systems that give you the opportunity to test for molecules or interventions, any type of small molecule drug, like the type that you end up taking as a pill that's in your medicine cabinet, or an injectable antibody for folks who take something like Enbrel for rheumatoid arthritis and psoriasis, that type of thing. But this is the process of finding molecules that have potential treatments. Then, after you've found potential molecules, then you go through the lead optimization step. So in the lead optimization step, you're trying to sift through the promising ones to find the ones that have the best parameters, right? The ones that are going to, if you're trying to find a drug for ALS and you're trying to protect neurons, so drugs that might get into the brain and into the spinal cord, drugs that are less likely to cause liver toxicity or to cause other side effects, right? Or blood toxicity. That's the lead optimization stage until you identify your lead, your one molecule that you really want to move forward. Then you go into this stage called IND enabling. The IND enabling stage, IND stands for Investigational New Drug. And this is a filing that you submit to regulatory agencies like the FDA, where you have demonstrated through experiments that your drug product, the thing that you're actually going to try to put into a clinical trial, is reproducible. It's always made of the same exact thing. It behaves predictably when you put it into a larger animal, maybe like a pig. And then once you've completed those studies and you file it, then you get to move into clinical trials. Now, the ALS clinical trial space right now is as full as it has ever been. There are many, many drugs in clinical development that are being tested in either phase 1, phase 2, or phase 3 trials, right? So phase 1 trials are the ones that are just assessing basic safety of this molecule. Does it cause harm when you put it into people? Once you learn that it doesn't, then you can move to a phase 2 trial, which is bigger and starts to look, it still looks for safety primarily, but it starts to then look for indications of efficacy, that the drug is showing trends toward benefit in people. And you may try to use these trials to identify the right people to test in maybe a phase 3 trial. And then finally, a phase 3 trial is typically the one which would be definitive. It gives you your final result that shows that the drug either helped or didn't help. And if it did help, then you get to your FDA approval. And that's a typical model. And I'll say this model for many diseases has worked, right? This model has produced antibiotics that treat infections. It has produced vaccines that have eradicated diseases. It has produced treatments that have decreased heart disease, and it has produced treatments that have been curative against some cancers. This process can be effective. There is value in being flexible in this process, and I think we're learning more and more in rare diseases that flexibility is incredibly important in this space, but there's still a lot to be done using this model and getting to good, safe, effective treatments for people with ALS. So now, where does ALS TDI fit in this space specifically? I like to think of us as a discovery engine, right? So those are my little gears. Carolyn Rogers made them the right color for me. So we are a discovery engine whose objective is to discover and invent new treatments for ALS over and over and over again until the needs of everybody with ALS are met. We have experience in having done this, and we intend to do a lot more of it. So we focus on target discovery. We focus on the process of inventing screens, inventing models, systems in which to test drugs. And we can do that in induced pluripotent stem cells from humans. We can do that in cell lines that have existed for a long time that act a lot like neurons. We can do that in mice, in mouse models of ALS. We can do that in zebrafish. We build systems in which to test and assess drugs, which will lead to the most promising ones, through which we conduct lead optimization to find the best ones with the best properties, which we then move and manage through the IND enabling process. So everything in here happens within the four walls of ALS TDI that I'm sitting in right now. Everything that happens here. We're never going to have a medicinal chemistry manufacturing plant, right? That's not something we're going to build at ALS TDI. So that's something where we have to find a partner, work with them, and we pay them to execute that work. But this here is done on site by the 30 scientists in the lab to my right of my door over here. So some examples of how this has played out for us in the past. We've done this independently, right, with AT-1501 in the past, where we discovered the target ourselves through the fundamental research. CD40 ligand was our target. We went through the process of testing therapeutics in animal models, and we identified a molecule targeting CD40 ligand that worked. And then we had to invent the drug for humans and optimize it to find the best potential molecule. And then we went through all of the IND enabling work, right, to manufacture that and tested it in larger animals before partnering with a for-profit partner and hospitals, right, the clinics. So multiple clinics across the country are running our phase 2 trial of AT-1501 right now, or Eledon's, our partner's trial of AT-1501 right now in people with ALS. So the process starts here, we hand it off, and it moves forward into the clinic where it gets tested, and hopefully, if all goes well, ends up being an approved option for ALS. We will see. Another example is different. AP101 was a partnership with a company called Neuroimmune. We did not invent that drug, and we did not discover its target, misfolded SOD1, right? That target already existed. And Neuroimmune, the company, the partner, had invented multiple molecules. We helped them figure out the lead. We did the lead optimization work with them, testing multiple molecules in our models to identify one, which they took through IND enabling studies and is now in phase. Actually, I think it just read out a successful phase 1 trial out of Canada about 3 weeks ago, and will be moving forward into phase 2. So that's another example. It doesn't have to be us. We will work with many other groups to identify promising molecules and help to move it along. And then we have this set of molecules right now, which are in our preclinical pipeline, where we have a lead set of molecules that were identified from an induced pluripotent stem cell assay and has been going through animal models right now. And so we're moving toward identifying a lead, which would then move into the IND enabling space. A little bit earlier on in the process, we're working on PRMT inhibitors targeting C9orf72 specific biology. We're still working on the drug discovery phase. We're still working on the lead optimization phase, and then we hope to move that as well into IND enabling. And at the same time, the engine that started this whole process is still in process to discover new targets and new leads to move them along. I hope this has given you a sense of who we are and where we sit in this process, and how important it is, because we know we'll need many treatments to address the needs of people with ALS. And so this is just a smattering of some of the team members who do all the work. These days, I just do a lot of talking. I talk to the team members. We go through the data together. We work on experimental designs together and the scientific strategy. But they're the folks who do all of the work. And I'm incredibly proud to be a part of this team. And with that, I'll now turn it over to Carol to send some questions my way and see if I can address questions from the community.
Awesome. And Fernando, I'm going to start you off with one that just came to my mind because I think it would be helpful for everyone to kind of understand this dynamic even better. The collaboration with Neuroimmune. So basically, please elaborate or correct where I'm wrong. Let's help everybody to understand. This was a company that had something that they thought might look good for ALS. How did they then come to us, to ALS TDI, and why?
I actually think I went to them
Okay.
on this one. This was a few years back. We had a program looking at SOD1 biology here at ALS TDI, and we were thinking about ways that we could use our learnings to advance treatments for ALS. And we had one internal program that was trying to find small molecule drugs that would stop SOD1 from misfolding. And we wanted a different approach using an antibody, right? A protein biologic that could also do it. So we wanted to attack it from two different angles. The small molecule approach didn't work, right? But at the same time, I looked for people who were in the space who had technology that could be brought to bear, so this type of antibody, and found them, reached out. We talked about our mutual goals and our respective capabilities. They had the drug, we had the in-house preclinical pharmacology. And it made all of the sense in the world for them to share multiple of their antibodies with us for us to evaluate in animal models here. So they didn't have to build that whole apparatus on site. They didn't have to hire a whole team. We could do that work for them and share those data directly with them. And it helped them to have the data to justify advancement into clinical trial.
Awesome. I love that story just because I think it's a great example of how everybody can increase efficiencies, spend less money in a space where there's limited resources, and move something forward into people, which is what happened here. So I think that's great. Fernando, this is kind of off topic from some of the things we've been talking about, but I know it's important to a lot of people on here. Are there any diets that have a connection to ALS, or any type of diet that has background to say that it may slow the disease?
So that's a great question. There is no specific diet that I'm aware of that has a connection to driving ALS, to causing it, or to slowing it down. What we do know is that high calorie, maintaining body weight, we know maintaining body weight is important, and patients who maintain body weight best have better outcomes, longer lives. In particular, for folks who are faster progressors, those who can manage to continue to keep a high calorie diet and maintain weight will end up having their progression level out to some extent. There are some studies that have demonstrated that. There have been a few studies in the past few years. So no specific diets, keep getting your calories in, doing whatever you can. There's a lot of evidence suggesting that people with ALS are hypermetabolic, meaning they burn through more calories to be alive every day. So you need to consume more to maintain weight, and maintaining weight leads to good outcomes.
Awesome. I'm also going to ask everybody, if you have questions, to please put them in the chat and have them go to all. I'm not able to monitor the chat as frequently as I'd like to. There are some that I see that are private messages to me in there, which are great, and some are very funny, and thank you. And those don't necessarily need to go to all, certainly. But if you do have questions, I do want to make sure that we record them so that we get back to you if we're unable to answer them. I don't see any follow-up this moment on the diet questions, so thank you. And again, we can provide follow-up as a team. And the next one, Fernando, is pretty cool. And I look forward to your answer on this one, and it's in depth. What are the prospects for antisense oligonucleotide therapies in ALS, ASOs, in light of the setbacks reported for the 2 trials in Huntington's disease, treatment of adult patients living with spinal muscular atrophy, SMA, and ongoing ALS trials?
That's a fantastic question.
It's a big question.
It is. So thinking about ASOs, you have to think about what its job is. So an antisense oligonucleotide, what is it supposed to do? An ASO is designed to, so we'll start with canon of biology. DNA leads to RNA, which leads to protein. So an ASO is a molecule that's designed to essentially stick to a specific RNA, which would have yielded production of a certain protein, so that the RNA goes away and that protein never gets made, right? So you're reducing the amount of something usually. There are some exceptions to this, but usually using an ASO, you're reducing something's amount. Now, that leads one to ask a question. When you're trying to develop an ASO for a disease like ALS, particularly versions of ALS that are caused by genetic mutations, right? So we all know, well, many of us know, that mutations in SOD1, SOD1, cause ALS. There's a lot of evidence around SOD1 demonstrating that when it's mutated, it changes its shape, it misfolds, and then the misfolded version causes some kind of havoc in the cell. It does something it's not supposed to do that's poisonous. So reducing the poison makes a lot of sense, right? That's a gain of function. The mutation is a gain of function. It makes the protein do a new bad thing. So that's poisonous. So bring that down. Sometimes the mutation is a loss of function, right? So their gene is mutated, the protein no longer can do its normal job. So it's less helpful in that context to reduce the production using an ASO, right? And that might actually make things worse. So sometimes you want to understand the biology of the mutation before you come in with an ASO to reduce something's levels. Now, there are some areas where we don't know right now if it's gain of function or loss of function. And so a lot of people are willing to take a shot and say, well, it's a coin flip, so let's try an ASO and see if we make things better. There's a risk we make things worse, but plenty of people with ALS are willing to take that risk. And I understand that, as long as they all understand. So I'll summarize by saying this. I think there's massive potential for ASOs. They likely won't work for every version of ALS, right? Even every version of familial ALS. We need to see how that plays out. But I'm very, very glad that the technology exists. I'm glad it's being tried. I I wish Sean was alive to have received the current SOD1 ASO that's in trial, right? That's how I feel about its potential. I think that would have been a good thing for him to try. So I hope that gives you some context. As far as I was surprised by the Huntington's results, I had been similarly positive about the ASO against the polyQ mutation in Huntington. So that shows you that there are many, many good ideas. They just aren't always right. And that might be one.
So Fernando, I just want to make sure too that everybody knows kind of why this question was posed and why it's important and timely. There are multiple trials out of Biogen and Ionis right now in ASOs in 2 different mutations and an early program in sporadic ALS. So this kind of category of drug is one that has been exciting and filled with hope. So Fernando, when the Huntington's trial failed, do you think that that said more about the mutation it was aimed at or the type or ASO as a drug?
I thought it said more about the mutation. I might be wrong about that.
Well, that's good news, right?
I don't think it says much about the ASO, right? I think ASOs, Ionis and others have made a lot of progress in understanding how to deliver these. It's just more about when it's the right thing to do, and when the mutation can be wrangled by this approach versus others.
And with ASOs, you talked about them most likely not being able to work for every single type of ALS. If they worked for a type of ALS, would they work forever for that type of ALS?
Unclear. It's unclear how it would play over time. We'll find out, hopefully, with something like SOD1, right? So we don't know if the body would eventually compensate and go into overdrive to try to produce more of the thing we're suppressing. These are all open questions, and time will tell.
And why we have to keep going, right? Because we've got to hedge those bets. Okay, so somebody asked why ALS is expected to increase 70% by the year 2040, which I had not seen that. So I'm intrigued by where that stat came from, if you've heard it, and what you think the answer is.
So I have not seen a reference for that specific stat. I think it's reasonable to expect that ALS will increase essentially because of an aging population, right? So more people are living longer, and age is the greatest risk factor for ALS. So you're likely to see more people with ALS. There is some evidence, though not confirmed, that the actual incidence of ALS in younger people might be increasing as well. I'm not sure if that's because diagnostic criteria are getting clearer and people are doing a better job of calling ALS correctly what it is, or if there actually are factors that are increasing the amount of ALS regardless of age. But I think the biggest contribution to an increase would be an overall aging population.
That makes sense. And the diagnostics too, I suppose, again, I hadn't seen that stat either, but I suppose also in the next, 18 years, we may have even better diagnostics, which by virtue of that could increase it too. So I don't know if they're anticipating that. Okay, you ready? We've got a specific one.
We'll see.
And I'm going to read it, really read it. In early May of 2021, results of a phase 2, and boy, I already know some of the conversation that's going to take place around this. Results of a phase 2 Italian guanabens study, the name of the study was PROMISE, were released that found the drug to slow ALS progression. An internet search of this person found that ALS TDI did a study on guanabens in 2015 or so, which is true. But the study showed that the drug was ineffective for ALS. Could you please discuss the two study designs and what made guanabens work in this phase 2 study? What does the recent news mean for the future of guanabens in ALS? And also, is it available for patients to try on their own? And is TDI expecting to do any more work on guanabens?
Okay, hold on.
I'll read. You can just start and I'll make sure you get the rest of it.
All right, so I've seen those results. That's an awesome question. Whoever asked that, thanks for diving in deep. Okay, so that phase 2 study, I would hesitate to say that guanabens worked in that study. That's my starting point. So the study, if I'm remembering the design correctly, there was a placebo group, a 16 milligrams per day group, a 32 milligrams per day group, and a 64 milligrams per day group. And when they looked at all of these people at the end of the study, and then they parsed out the ones who had bulbar onset only, right? And there were only a few people with bulbar onset in each one. They said that the people in the high dose, right, the 64 mg dose, did not have the same level of decline as the people in the placebo group. Now, that's a lot of parsing to find that specific effect, right? And the numbers ended up being very, very small to see that. So I hesitate to call that like where the drug worked. I should rewind for a second and explain what guanabens is. Guanabens is a drug that's used for hypertension in some cases, so high blood pressure for some people. In around 2011, a paper came out that said that the drug does more than the blood pressure thing, right? The drug actually, if you put it in cells and you give the cells a stress that looks a lot like ALS, it's called endoplasmic reticulum stress, and then you treat those cells that are stressed with guanabens, it protects them. And we actually did that work too. We tried to replicate that work, and we did. In the cells, it does protect them from that endoplasmic reticulum stress. So we took that, we were excited about that. It's like, great, published results, we replicated it. That doesn't always happen, so we're feeling great. Then we tried it in our mouse model, and then it's revealed in the mouse model that the drug is still a blood pressure drug. It's still an alpha-2 adrenergic receptor agonist, and that does all kinds of other things, right? And we actually found that the other things that it does probably makes mice worse, right? Being an alpha-2 adrenergic receptor agonist. So it makes it a not great drug for ALS. It was not beneficial. I actually think those findings are quite consistent with this trial, where if you read the entire paper, they actually talk about increased adverse events in people who were on the drug compared to placebo. It was markedly higher. Wow. And in the conclusion of the whole paper, they don't advocate for advancing with guanabens. They advocate for advancing with a drug that is like guanabens, but doesn't have alpha-2 adrenergic receptor agonist effects, right? So guanabens minus that other stuff, which is consistent with our finding. So I think it's interesting. As far as what we will do, we're done with guana bends for the same reason I just described. We have had other work ongoing around molecules, though, that also don't have the alpha-2 adrenergic receptor agonism. And we're actually Theo, Dr. Hatzapetros, who's often on the other side of my wall here, he's been working on writing that up to share those details with the community as well. So I know that that was all a mouthful. It was a great question. Our results are not inconsistent with the trial results. I just think they might be inconsistent with the headline, the title of the trial result.
Yeah, that makes sense. And so conceptually, first of all, when you say they didn't recommend moving forward with guanabens, but with a guanabens minus lots of multisyllabic word agonists, they would recommend. Is that a thing or is that conceptual? Like, is there a drug standing out there waiting?
No, that needs to be I mean, there are molecules like that, but there's nothing approved like that. Okay. Right? So it's still, if you're talking about where it is in the pipeline, that's in kind of like the lead optimization moment.
And you also referenced the sample size of this phase 2 in Italy. I know this is a topic that we try to talk about as much as we can, but there are some people on here that I know have not been on at least our town halls in the past. Could you talk a little bit about why sample size matters, especially in a disease with this level of heterogeneity?
Yeah. So it's a little bit easier for me maybe to describe it when you're doing mouse studies, right? So imagine you have a mouse study. You can control everything, right? Like these mice are basically all little twins of one another. If not twins, they're family members, right? Quite related, and their disease is really quite similar. If you don't have enough animals in a study, it's very easy to fool yourself into thinking that a drug either makes the mice live longer or live too short of a period of time. There's the variability, the randomness of how a person or an animal can progress can make it really hard to not trick yourself. So you can get around that by either having really, really stringent selection criteria, like we do with our mouse studies. We make sure that our selection criteria include like really careful assessment of their genomes, making sure that we have the same number of male mice and female mice, making sure that mice from the same family don't get all thrown into the same group because they tend to live to be about the same age. You could imagine doing something similar in human trials if we had the right selection criteria.
Yeah.
If we knew how to parse folks so that we could make it, that we could measure something with a really small number. We're not there yet, so numbers need to still be relatively big, at least for sporadic ALS studies.
Yeah, that's a tough one. And it kind of leads into this. This is conceptual and could be a really cool, sitting Indian style on the floor with a glass of wine for hours conversation, I bet. Would a consensus regarding the pathomechanisms of ALS provide more opportunities to explore different avenues of treatment?
It all depends on if the consensus is right.
Yeah.
Right. So,
but how do you even develop that consensus?
Yeah, it's so I think that can happen over time, right? That does happen in science over time. You do develop a consensus. Like you find out new things, you update your model, you find out something was wrong, and you gradually triangulate toward truth. That's what we're trying to do. We're not there, right? There are a million and one ideas in the ALS space for therapeutic approaches that might have value or what's driving the disease. Um, some are better ideas than others. Some have more evidence and support than others. Um, I said it earlier. I don't know if anybody noticed. Lots of great ideas are just wrong, right? Like there are great ideas. You can line them up. You can be as clever as you want to be. And the reality is you just don't know something really important that if you knew it, you would realize you were completely wrong. But in the meantime, you've created this model that makes all the sense in the world to try to test. Um, so we're not in a position right now where we're close to consensus on, you know, a pathomechanism because there probably isn't one.
Yeah.
Any right? The reality isn't that. Um, if we did have one, I don't know if it would allow us to explore different avenues. I think it might actually hone in this hypothetical world where there was consensus that was right around pathomechanism. You might just try different ways to tweak that target space most effectively, and it would more narrow things, but maybe make you more productive. I hope that answers it. That's a big one.
Yeah, it's a really big one. I think that's the beginning of a comprehensive answer, because I think the important thing is a consensus can still be wrong.
Yeah.
Okay, this is a specific one, but one I know that matters to a lot of people. This was a hot topic in at least the ALS community social media realm a month or so ago, and it's about NU9. Wondering if you have heard of NU9, which the answer is yes. Found at Northwestern, and this person felt that it looked so promising for rejuvenating neurons. And could you give us your thoughts on that?
Sure. It was a really nice paper, and it demonstrates there is potential for NU9 to become a therapeutic with a good bit of work. And I think Dr. Osnler and Silverman are working toward. There's all of those steps that you have to go through, right? So there's still probably lead optimization that might not be the perfect molecule, and they would still have to go through all the IND enabling steps. There's a lot of work there. What I guess I want to mention is there was a heavy emphasis on looking at upper motor neurons, right? And I think that's good. It's a useful approach. Upper motor neurons are the ones in your brain. Lower motor neurons are the ones in your brainstem through your spinal cord. Upper motor neurons connect to lower motor neurons, and lower motor neurons connect to your muscles all over your body. And for ALS to be ALS, you have to have impairment of both upper and lower. And I think it's fair to say that ALS research has often focused on the spinal cord and lower because the models have typically presented with more severe symptoms in their lower motor neurons. So the most interesting and different thing about the NU9 study to me was a focus on protecting the neurons in the cortex. And they make a point that their molecule does that. They make the point that their molecule is the first to do that, which I'm not sure is true because people haven't looked with other molecules, right? So we didn't look to see if CD40, if anti-CD40 ligand or AT1501 protected upper motor neurons. It may have. I don't think the folks who did the work on the SOD1 ASO looked to see if the ASO against SOD protected the upper motor neurons. It probably would have. So that's a caveat to that statement that it's the first ever to do that. You can only see what you look for, essentially. And
Yeah.
that's another important thing to remember in research.
So, so basically, I think it's NU9 might be a little earlier than people originally thought when you think of that drug development pipeline that you put up.
Maybe. I don't know the details of
Yeah.
where they are, but I think it might be a little earlier than people initially thought. Yeah.
And then Fernando, help me with this. And maybe, I'm a good example of things other people are thinking about, hopefully, are upper motor neurons not upstream of lower motor neurons? So is it more of a grand slam if you're protecting upper motor neurons, which are then connecting to lower motor neurons?
We don't know. Speaking of lack of consensus, and the two things might be true for different people. We don't know if it's a die forward, right? So from motor neurons to upper motor to lower, and then out to the muscle, or if it's die back, right? So if it's lower motor neuron letting go at the neuromuscular junction, at the connection to the muscle, and then dying and then resulting in upper motor dysfunction. Now, what we know is that different people with ALS manifest with different symptoms, both upper and lower at different times. So it may vary for each person. So I wouldn't say, though, it's more of a grand slam. I would say it's equally important.
Okay. So actually, the next question that we received is about why symptoms vary so much from patient to patient and why the disease is so difficult to diagnose. So great segue here.
So we don't know why symptoms vary from patient to patient. That's one of the more challenging mysteries and frustrating ones, right? Like going to my friend Sean's case, right? He had the same mutation his mom had, right? His disease hit him in his 30s, and it hit his mom in her late 50s, and we have no idea why. At the same time, something like C9orf72, those mutations, you can have the same family. One person's disease will manifest as ALS, and another person's disease will manifest as frontotemporal dementia. Completely different diseases caused by the same mutation. If we could understand what makes one person's type of neurons more susceptible or more resilient, those are the types of sort of translational research, so direct from patient to bench types of research that give you the opportunity to learn about targets. Like, so if we could figure out why one person's motor neurons don't succumb to the C9orf72 mutation, maybe we could make everybody's motor neurons do that in the face of C9 or in the face of other pathologies that cause sporadic ALS. So these mysteries are frustrating and they make it hard for us to immediately develop treatments, but they're also examples of the opportunity in what is unknown. Because in all of this space is where we can discover the ways to intervene.
So many ways to potentially intervene, though, right?
Yeah.
We have that makes it really hard.
Yeah.
So also, Fernando, like when you talk about Sean and his mother, which I remember, and I have some friends in New Jersey who had a similarly aggressive form of SOD1 ALS, and they lost a mother in her 40s and there were sons in their 20s who got the disease. But we also see, variation from, slow progressing, fast progressing, where the disease onset is, both limb, bulbar, hand, whatever. We've talked about, there's been conversations over the years around predispositions and neurotoxic triggers. Do you think that multiple combinations of what those can be have an effect?
Hmm. So there's a hypothesis bouncing around that there's like a number of steps that need to be tipped for a person to have ALS onset. And this, I think, Dr. Amar Al-Chalabi has been on sort of the cusp of this. And one of the things they've demonstrated is it's like a mathematical function where it seems like people with dominant mutations, so familial ALS, end up having one less step, right? So the mutation is
There and ready to go.
yeah. And then the other 5 steps, all you need is 5 instead of 6 is kind of their operating hypothesis. I think there may be some truth to that. The way I think about the ALS process, right? And bear with me, this is an analogy. I actually said it the other night at the ALS research paper review thing that I did last Thursday. I think of the ALS pathogenesis as a series of interlocking dominoes sort of set up in a room in rings that are kind of connected up to each other. And you can tip them at multiple spots, and eventually they can all topple. And it doesn't necessarily matter which one or which order, but if more of them get tipped at once, more will come down faster. And if only one gets tipped, then maybe they don't all come down faster. And if you pluck dominoes out at the right spots in those rings, maybe you stop the process altogether. And that's kind of how I picture the problem, where lots of different spots for things to go wrong, but then lots of spots for things to go right as well.
So we want to pick the dominoes to pluck out.
Yes.
In the analogy.
Yep.
So when can we figure out which dominoes to pluck out, Fernando? That's actually the next question is, when can we expect a cure?
Well, so
I love the brazenness of that question. Thank you very much.
Awesome. And I will have a brazen answer.
Great.
There won't be a cure. There will be cures. I believe there will be multiple cures, right, for different types of ALS, but there won't be a cure for all. I think there will definitely be effective treatments, which in combination could be effective cures, right?
How do you differentiate between those two things, an effective treatment and a cure?
Well, it depends on where you are in your disease, right? So a person who is just now having issues with, their right hand, if there's a treatment that they get quickly enough that effectively halts the disease and they recover, 80% of that function, I would qualify for that person, they were largely cured, right? A same treatment that halts progression for a person that's, has an FRS score of 27, that's not a cure, right? It's a treatment. It's keeping them from getting worse, but it's not a cure. So the same treatment might be a cure for one, but it's a treatment for another. What we like to think about as a cure altogether, like a complete cure, a panacea, would be one that halts progression and then you recover all function. I think we are far from that.
Do you think it's possible someday?
Yes, I think it's possible. I think it would require, technological advancement that is hard to put the dominoes, like line up the pieces that would say, here's how you get there. But it would require halting disease and then regeneration. Yeah. And we're far from both. When you think about regenerating, you're asking a neuron to grow 3 feet and go from your back and sprout all the way down to a muscle in your foot. And I think we're far from that type of regenerative technology, but breakthroughs happen. So
Well, and I think, let's take let's work on effective treatments so that everybody who is on this Zoom right now, we can keep them alive for that breakthrough. That's
yes, that's the
what I would like. And as we talk about, again, really so much of what we've been talking about, kind of circles around the heterogeneity of ALS. And you and Steve Perrin and, so much of our team back in 2014 put together the concept of a precision medicine program in ALS, which has just been, incredible. And it still astounds me, quite honestly, that it really, that no one was ever doing this. It's really quite amazing. We'll probably talk a little bit about the ALS precision medicine program now, since I'm about to ask a question about it. But if you're on here and you need more, please, ask any of us after this is over, because it's pretty darn cool. But somebody asked about it, saying, what are the specific advancements in understanding and treating ALS that have come out of the PMP, ALS Precision Medicine Program? And what's the status of the program? And have you amended goals around it at all?
That's a great question. The Precision Medicine Program, we call it the PMP for short, was a really audacious program. It was Steve Perrin's brainchild. And he said, you know what? Whole genome sequencing has become more affordable. There's this whole new type of technology called induced pluripotent stem cells that you can actually do something with in the lab. It's matured to the point where you might be able to do something with those cells. And there's this whole realm of digital technologies for tracking disease. Maybe we can do better with that. And let's do it all at once. Let's get everything we can, and let's go directly to the patient community, right? Instead of setting up multiple IRB protocols, institutional review board protocols through lots of different clinics and things like that. We have this connection to people with ALS. We think they would join this and they would contribute. And he was really well ahead of his time. Lots of folks have since kind of copied this, and to good effect. I think like the entire community is moving forward because lots of people are doing this type of work, but it was Steve's idea. And as far as specifics, that accomplishments, one accomplishment, and we haven't published these results yet, that's in process. Dr. Kyle Denton here is working hard on that, is we have used induced pluripotent stem cells as a result of the precision medicine program, specifically one cell line where the neurons do get sick. They're ALS related neurons and they get sick. They die sooner than normal neurons do. And we've exploited that cell line to test drugs. So we're testing a large compound library, 36,000 compounds, plus a few targeted screens within that as well, to find molecules that have potential as treatments. And we have identified a lead series from that effort, which they've also worked in our animal model. And that's actually one of our most important programs. So a tangible output from the PMP are drugs we're inventing. On the clinical side, so clinical outputs, we did collect data using accelerometers and voice recordings. And we've worked with Google to turn those data into quantitative measures, right? So a way to count how a person progresses in a way that we believe is more objective than the functional rating scale survey. And we have a paper that's in the process of review right now, where we describe all of those results. As far as there was a next direction, next goals, right? That's,
Yep.
yeah. Um, a really big push right now is building off of what we initiated in 2019, which is the in-home blood collection program, where our participants, about 100 right now, are giving quarterly blood draws. So a blood sample or a number of blood samples every 3 months. And we're going to work hard to look for blood-based biomarkers that would predict progression and would demonstrate when a drug might be working. And not just generalized biomarkers, we also need biomarkers that are specific to our drug discovery programs, right? So, for example, in the case of the C9orf72 research program, we want a biomarker that would demonstrate that the drug we're testing, a PRMT inhibitor, right, is having an effect on that specific target. And that would help us hone in on dosing. So it's kind of a holistic view of how to use biomarkers. And then finally, a big piece of, as far as direction goes, is data sharing. Like a really big effort to share these data, get more eyes on them in the research community, because we think that can help move things along faster.
So I think the biomarker stuff and the blood collection is really cool because that was a change, and an addition after the beginnings of the program. So what I really think is cool is that as we learn more about what we need to learn, you all will amend the program to find a way to learn it, which you did in this case. And I like that you just talked about, that there are different types of biomarkers there. Biomarkers of progression, there are drug response biomarkers, which you're talking about with C9, which is also a great lead-in because both in the chat a minute ago and in some of our other submitted questions in the chat, somebody asked, why are there so few when C9 is quite prevalent, especially within the realm of familial genetic ALS? Why are there so few C9 studies? And someone else asked that could we get an update on the work that TDI is doing specifically in C9?
So why are there so few C9 studies? Gosh, to me, there aren't so few. There's a lot of fantastic work being done in C9orf72 biology at the research and drug discovery level.
Well, see, I think it's important, I'm glad you said that because sorry to, cut you off mid-syllable, but very often in the ALS community, listen, silos are real in research, right? Just going to say that. But there are fewer than I think everybody thinks there are. Mm-hmm. Often someone will say, hey, has Dr. Vieira heard about this? And he heard about it, 2 years ago while it was still a glint in the researcher's eye. So there are programs going on in C9 that the rest of us don't even know about, that you know about. Mm-hmm. So I'm glad to hear that. Yeah. So you feel like we are learning, so I know C9 itself is more complicated than we would like it to be, right? Why does the higher power do that where it's the prevalent mutation, expanded repeat, and we're going to give you the big one, but we're going to make it the hardest one of all. So you feel like we are learning more about that, and with each bit, other people are learning and contributing to the knowledge sphere. you're expanding TDI's work in C9, or how did you decide to kind of invest in C9? And how is everyone's work bettering ours in there?
So we decided to work on C9. It was an obligation, right? It's the most common known cause, right? So um, I understand it's still in the grand scheme a relatively small proportion, but we know something about it, just like we knew something about SOD1. So for years, we had a really robust SOD1 program, and where we built internal capability and we worked hard on that neuroimmune collaboration. And the objective was to help deliver a treatment into clinical development. We accomplished that goal. And then we looked at the landscape of SOD1 and recognized that a lot of folks were doing a lot of work. Ionis and Biogen were working on ASOs. Voyager was working on gene therapies. A lot of work was coming out of UMass Medical. They were doing a lot of work, and there were drugs moving toward the pipeline in the SOD1 space. And we said, C9orf72 is more prevalent. We can make more of an impact if we take the effort that we're putting in SOD1 now that we delivered on producing a lead molecule and invested into C9orf72. C9 is a complicated type of mutation, complicated biology, like you alluded to. There's a specific lane of that we focused on, which are the dipeptide repeat proteins, largely because it wasn't too foreign from the lane that we had worked on with SOD1, looking at misfolded proteins or strange proteins. And so we decided to go all in on that, build off of existing literature that was coming out from the research community. We validated some previous work and built on it, and then built a system to test drugs here. And that's how we got to discovering a whole new target. But it is an obligation. There are so many people living with C9 mutations. You want to sink your teeth into the pieces where you know a lot.
Yeah.
And in C9, we knew more than we knew about other types of ALS.
And it has the opportunity to help a lot of people.
Yeah.
You know, I mean, Fernando and everybody, we have to, you know, start to wrap up here, but this is a question of my own. We talked about, you know, is there a trend towards, you know, people getting diagnosed more frequently? C9, I would say every day I get a call about someone else with C9. But that wasn't the case immediately after the discovery of its relevance. Is that because now it's included in more genetic panels and people are discovering the mutation more? What are you finding that, and what do you think that's all about?
I think so. It is because you can now test for it. Testing for the C9 mutation is different than testing for most mutations. Most mutations are like a spell check, where you look for a misspelled word. Looking for this mutation is looking for a correctly spelled word that occurs over and over and over again. So you had to do it a different way. And immediately after the discovery, that assay, that system for screening didn't exist for a few years. So now it's there. I think for a long time, people who had this type of familial ALS just didn't know the mutation, right? They just knew that there was this scourge in their family.
Yeah.
Or some of the people ended up having what they thought were Alzheimer's.
Yeah.
And then you'd have a random sporadic ALS patient, right, in that family, and people missed it. Now the community is screening more widely, so we're identifying it much more.
Now, and just to close, because I think this is an important point, do you believe that discoveries, no matter where they are in the drug development pipeline, or continuum, let's say in this case. Discoveries regarding C9, SOD1 plus TDP43, will be relevant to sporadic ALS.
I do. I think we learn about the biology in each of these domains, each of these types of mutations, and that there will be overlap and sort of the Venn diagram of processes that are in play in sporadic ALS. And you might have a sporadic patient whose disease looks a bit more like C9. You might have a different sporadic patient whose disease
committee to actually fund some of this research through the defense.
I do think that it's highly relevant to sporadic ALS.
Okay, awesome. I just want to make sure that everybody always hears that. No one is forgotten for one moment in the work that we are doing. Okay. Hey, ALS TDI team, we have a lot of work getting back to answer all of the questions that are in the chat. And there's still some outstanding ones that were submitted with registration. There are some that are coming into my phone, and I imagine other phones across TDI. So give us some time, or pester us, either one, it's fine. We want to get you the answers to your questions, because that's the entire reason we're doing this. Remember to answer the survey, give us honest feedback, criticize us when necessary. If you have suggestions, don't just criticize, give us an idea of what to do better. And we will try, because, we're not doing this for us alone. We're doing this because we are part of a community, and there are certain people like Fernando who have the skills to be able to make this community grow and live longer and enjoy each other's company for years and years and years and generations to come. So we need your help with this. Bill Hassel, this entire time, I thought that was a still photograph because you looked so perfect. And look at you with the accelerometer on your wrist. God bless you. Thank you, everybody. I'm sorry we couldn't get to every question. All of us here are delighted to have Fernando as our CEO. I've worked with Fernando for 16 and a half years. You got to see a little bit of the side of him that shows you that his heart is as big as his brain, is as big as his commitment to ending this disease. So thank you, Fernando. Thank you, everybody who is on here. And we look forward to following up with all of your questions. Have a great day, everybody.
Thank you. Thank you all.